Hi everybody, I'm Katie Couric. This is Eye to Eye. Now it sounds like a Frankenstein fantasy, but the science of growing organ tissue is on the verge of some unbelievable breakthrough. Today we present you with a video that takes you through what you may call the lab of the future.
It may sound like science fiction, but researchers at the University of Minnesota say they've grown an artificial heart. Scientists started by removing the cells from the heart from a rat, seen in this video provided by the university. That left the outer structure or scaffolding. Then they injected new heart cells from newborn rats. In 2 weeks, the cells grew around the scaffolding, creating a new beating heart.
Need a new heart or liver? Science is making spare parts for humans.
The whole idea of tissue engineering is how do you get those cells to actually form the tissue and to carry out the functions of tissue.
The directions for a human being are written in code.
Because the supply of donor organs just cannot keep up with demand, organ replacement has become a major goal. Well, first they get some cells.
Typically you have a donor material that may be the patient's own cells. Cells may also come from another source. The whole idea of tissue engineering is how do you get those cells to actually form the tissue and to carry out the functions of tissue.
Cells may have some memory of what they're supposed to do, but they also need structure, nutrients, and oxygen. Structure comes from what's called a scaffold, and they come in many, sizes. These are biodegradable polymer scaffolds for ears, and this is a scaffold for a finger.
The scaffold gives the cells a structure on which to grow. Without a scaffold, the cells are free-floating, they can't connect with each other, communicate, and form tissue. With the scaffold, they have structure that they need for a certain period of time until they've formed enough tissue to have their own structure.
When that happens, the scaffold dissolves. These are tissue-engineered ear cartilages for babies. Linda Griffith's lab at MIT has developed another kind of scaffold for liver regeneration, a liver chip. It's a tiny bioreactor that grows liver tissue from liver cells.
This is the heart of our whole system. The cells live on the silicon chip that you can see inside there. They live in little tunnels through the chip. And when we're growing the cells, it's hooked to a life support system that pumps culture medium in through here, and it flows across the surface of the chip, and it flows out this exit here. So this is essentially the equivalent of an artery in an organ, and this is the equivalent of a vein. Fluid comes in, gets consumed, oxygen and so forth, and then comes out here.
Now that you have seen what organ growth is all about, we will take you to our debate room where my colleague Francisco Rosales will host a discussion between different experts in the field.